Okay. And Brooke, can you see if you can share your screen?
Yeah, I have the option to.
So you want to get started?
Do you want me to start it?
Because your chapter is like the first one on the list.
Okay, cool. Yeah. Um, and everyone see my screen before I click start?
Yeah, we can see.
Okay. So hi guys. I'm gonna be doing chapter 12 and I believe part of 13. Correct me if I'm wrong. But anywho, to start off,
Oh, by the way, can you put in your slideshow in the chat?
yeah.
Sorry, thank you.
And just to make sure, can everyone still see my screen?
Yeah, you're good.
Okay. So to start off, I'm mainly going to talk about the nephron and basically the different parts of it, sodium reabsorption, water reabsorption, and everything such as that. So to start off, sodium reabsorption is active in most sections of the tubule, but passive in some. So the proximal tubule does 67% of filtering sodium. The ascending limb of the loop of Henle, remember that, does 25%. And sodium is not reabsorbed from the descending limb. This is a very important thing to know. And the distal tubule does 8%. Active steps involve the sodium potassium ATPase pump in the basolateral membrane, and the transport of sodium across the apical membranes is considered passive. With water reabsorption, water is passively reabsorbed through osmosis, and the ascending limb of the loop of Henle is impermeable to water. And reabsorption in the distal tubule and the collecting ducts is subject to hormonal control. Water passes through the aquaporins, also known as AQPs, and AQP1 channels in the proximal tubule are always opened, while AQP2 channels in the distal tubule and the collecting duct are regulated by vasopressin. We'll go over vasopressin a little bit more later on. This is an image this one you should recognize, I believe, from either the textbook or his PowerPoints. This one I found on the internet because I thought it was a little bit more helpful in understanding where the salts go versus where water goes, while the image on the right does a better job of explaining the different structures and where they are in relation to each other. So glucose and amino acids are also reabsorbed, and they are 100% reabsorbed, which reflects their nutritional value. And this is done through secondary active transport. So the symporter in the apical membrane simultaneously transports sodium down its concentration gradient. And then a specific organic molecule is then translated up its gradient from the lumen in the tubular cell. And due to this, the basolateral sodium potassium pump indirectly drives this co-transport system. And then once inside the cell, the organic molecule is transported into the extracellular fluid by facilitated diffusion. So what happens if blood glucose is too high? So this can happen because the plasma membrane carriers exhibit saturation, meaning there is a max value they can accept. So plasma glucose is high in diabetes mellitus, also known as hyperglycemia, and glucose is filtered into the Bowman's capsule at the same concentration as in plasma. And when the filtered glucose load exceeds TM or the tubular maximum for glucose reabsorption, the excess goes into the urine, and this first occurs at the renal threshold. And this is why for diabetes mellitus, glucose in urine is considered a diagnostic trait. So RAS, also known as the renin-angiotensin-aldosterone system. This is the regulation of plasma. Sodium is important for blood pressure regulation. And the granular cells of juxtaglomerular apparatus, or JGA, secrete renin into the blood in response to a fall in either sodium, extracellular fluid volume, or blood pressure. And the granular cells secrete renin in response to a fall in pressure in the afferent arteriole. The macula densa cells respond to a fall in distal tubular sodium chloride and stimulate renin secretion. And the baroreceptor reflex triggers increased sympathetic activity, which stimulates renin secretion. So in blood, renin catalyzes conversion of angiotensinogen, a circulating protein, into angiotensin 1. Angiotensin 1 is then turned into angiotensin 2 by angiotensin converting enzyme, also known as ACE, in the pulmonary circulation. Angiotensin 2 is the primary stimulus for aldosterone secretion from the adrenal cortex. Angiotensin 2 also stimulates thirst and salt hunger and constricts renal and systematic arterioles, increasing the blood pressure. So the effects of aldosterone include increasing sodium reabsorption by the distal tubules and collecting ducts, stimulating the secretion of vasopressin, which promotes water retention by the kidneys, and potent constrictor of systemic arterioles, which directly increases the blood pressure. So here's a little diagram that explains the effects of everything. I'm a visual person, so diagrams like this kind of helped me learn it better by showing what happens if something increases or decreases, and then what's the next step after that to continue on. So the natriuretic peptides oppose the RAS system. So atrial natriuretic peptide, AMP, is secreted by atrial cardiac muscle cells, while brain natriuretic peptide, BNP, is secreted by ventricular cardiac muscle cells. And natriuresis means increased urine excretion of sodium and H2O, increased glomerular filtration rate, inhibits sodium reabsorption in distal tubule and collecting ducts, inhibits secretion of renin, aldosterone, and vasopressin, and decreased cardiac output, and inhibits the sympathetic activity. So the kidneys and pH. So hydrogen ion secretion is important in regulating the acid-base balance. Hydrogen ions make things more acidic. And this takes place in the proximal, distal, and collecting segments using proton pumping VATPases. The extent of hydrogen secretion depends on the acidity of body fluids. When body fluids are too acidic, hydrogen secretion increases, and when body fluids are too basic, hydrogen secretion decreases. So the plasma potassium concentrations are tightly controlled. Elevated plasma potassium increases excitability of nerve and muscle tissues. It is actively reabsorbed in the proximal tubule and actively secreted in the distal tubule and the collecting duct. Most potassium in urine is derived from controlled potassium secretion in the distal parts of the nephron, and potassium secretion is coupled to sodium reabsorption by the basolateral sodium-potassium pump. And aldosterone stimulates potassium secretion and sodium reabsorption. So the countercurrent multiplier system. So the fluid in the proximal tubule is isotonic with blood plasma. This is approximately 300. And the close proximity and counter current flow between descending and ascending limbs allow important interactions between them. So sodium chloride is reabsorbed from the thick ascending limb by active transport, increasing the osmolarity of the interstitial fluid of the upper medulla. The solute concentration of the descending limb increases as water is attracted to the more concentrated interstitial fluid in the upper medulla. And a vertical osmotic gradient is established as the countercurrent flow of tubular fluid continues to deliver more sodium chloride into the medullary interstitial fluid. I know that's a lot of words and it sounds very, very complicated. That's why I include these images here. I, again, learned really, really well off of images. So I think it's better to just remember what increases as you go down and how the different osmolarities change when you're looking at the descending limb, ascending limb, collecting duct, and all of that. So the vasa recta. The vasa recta helps conserve the vertical osmotic gradient in the renal medulla, and the countercurrent exchange occurs when the vasa recta forms a hairpin loop. So this is theoretically what it would look like without the vasa recta that forms the hairpin loop. And here is what it actually looks like in our bodies, thanks to the hairpin loop and how this again affects the countercurrent exchange. So the extracellular fluid volume is most closely regulated to maintain blood pressure. A reduction in the extracellular fluid volume lowers blood pressure by decreasing plasma volume. Temporary relief of blood pressure disturbances is provided by the baroreceptor reflex, which increases/decreases in heart rate in response to pressure receptors in the aortic arch, the carotid artery, and fluid shifts between plasma and interstitial compartments. So water balance is regulated primarily by the kidneys and thirst. Free water reabsorption and excretion are regulated through vasopressin secretion. Water input is regulated by thirst. So the more thirsty you are, the more water you want to drink, which increases your input. And vasopressin secretion and thirst are both stimulated by hypertonic blood and suppressed by hypotonic blood. Osmoreceptors, the thirst center, and vasopressin secreting cells are located in the same area of the hypothalamus. And the left arterial volume receptors stimulate thirst and vasopressin secretion when blood pressure falls. Vasopressin promotes the retention of water and the increase of blood pressure. And here again is another little diagram that explains how thirst and vasopressin work together to increase blood pressure and what happens in the different routes you can take. Thank you. That's all I have for chapters 12 and 13.
Okay, now I'll start with the rest of chapter 13 and then we'll on to chapter 8. Let's see. Okay. Can you see my screen?
We can now.
Okay, good. So exam 3 review. So this is the rest of chapter 13. So let's start with responding to changes in pH balance. Let me get myself out of the way. Okay. So the respiratory system is the second line of defense that responds to the changes in pH in our bodies. It's not as fast as the body system, but it is faster than the changes by the excretory system. So when the arterial hydrogen ion concentration increases, the respiratory system is stimulated to increase the ventilation rate, which in turn excretes more CO2, which means more carbonium ion is available in the plasma, and the concentration of hydrogen ions is going to decrease. So this is going to kind of balance it back out. And then when arterial hydrogen concentration decreases instead, the rate of ventilations will also decrease, which will cause the body to use the metabolically produce carbon dioxide and add it into the bloodstream, which is going to in turn produce more acid and drive the pH levels back down. So that's how the respiratory system corrects it, just through adjusting the ventilation rate. And then, oh, okay. And then acid base imbalances. No, this is wrong. Okay. So the kidneys. Uh, the kidneys are the third line of defense, and this is the slowest method the body uses. And the change for the response to become evident, it requires hours or even days. And so the kidneys regulate both the hydrogen ion concentrations and the hydronium ion concentrations. Gill transporters, they can excrete both the hydrogen and the carbonium ions, but kidneys will simultaneously exchange the hydrogen ions for carbonium ions instead. And then in the kidneys, hydrogen ion excretion is achieved primarily by active transport and also by secondary active transport with sodium hydrogen ion pumps. No, not pumps, antiporters in the proximal tubule. Type A intercalated cells in the distal and collecting tubules are going to secrete H plus uh, and reabsorb potassium ions and hydronium ions. And this system, these cells are active at normal pH levels or when we have acidosis. Type B intercalated cells are going to reabsorb hydrogen ions and then secrete hydronium and potassium, and this is going to be active during alkalosis. And then so respiratory acidosis is due to hypoventilation and it's a buildup of carbon dioxide. To compensate for this, the kidneys will conserve HCO3 and excrete the hydrogen ion. And then respiratory alkalosis is the loss of CO2 due to hyperventilation. And to compensate for this, kidneys will do the reverse and conserve H plus and excrete HCO3 minus. And then metabolic acidosis is a reduction in plasma HCO3 minus, which with normal CO2 levels. And this happens due to loss of fluids rich in HCO3 minus or accumulation of non-carbonic acids in the bloodstream. And the most common causes are diarrhea, ketoacidosis, strenuous activity, and uremic acidosis. And to compensate for this, ventilation is increased. Kidneys will secrete more hydrogen ions and conserve more hydronium. And then metabolic alkalosis will is an increase in plasma levels of HCO3 minus with normal carbon dioxide. And this is due to a loss of fluids rich in hydrogen ions. The most common cause is vomiting, and to compensate for this, the reverse is done. Ventilation is reduced, and kidneys conserve H plus and excrete HCO3 minus. So essentially, acidosis and alkalosis, whenever the body's trying to correct either one of them, they'll have the opposite effects. And then environmental acidosis is increased carbon dioxide or other acids in the environment. So some forms are increased CO2 in the Earth's atmosphere because of how much fossil fuels we burn, or ocean acidification or sulfuric acid in the environment. And environmental alkalosis happens in highly alkaline bodies of water. Okay, so that was the end of chapter 13, and now chapter 8 on muscle physiology. There's three types of muscles, and the first one is skeletal muscles, which is attached to the bones. It is striated with long cylindrical and multinucleated structure. The skeletal muscle is controlled by neurogenic control by the somatic nervous system, so it's voluntary. And then contraction mechanism. This is like a really short overview because the skeletal muscle contraction mechanism is very detailed in the following slides. But calcium essentially binds to troponin. Troponin changes shape, and the binding sites are uncovered of the actin. And myosin globular heads will bind to actin. The filament will move, and the cycle is going to repeat. And then there are also the T tubules, which essentially help propagate the oxygen potential deep into the tissue. But yeah, I'll talk more about these soon. And then, so the other two types, cardiac muscle is found in the heart. It is also striated with branched mononucleated structure. The cells are connected by gap junctions, and this is myogenic. So the cardiac muscle can function without neural input, and it is regulated by the autonomic nervous system. So it's an involuntary muscle, and it functions similarly to skeletal muscle. But the calcium for cardiac muscle comes from extracellular sources and not just the sarcoplasmic reticulum. Smooth muscle is found on the walls of hollow organs arranged in sheets. It's non-striated with mononucleated structure, thick, thin, and intermediate filaments. It has thick, thin, and intermediate filaments. The cells are connected by gap junctions. Can be neurogenic or myogenic control. And it's also regulated by autonomic nervous system. So it's also an involuntary muscle. There are no sarcomeres in smooth muscle. Calcium comes from extracellular sources. The muscle contracts more slowly and uses less energy than skeletal muscle. And then, so in skeletal muscle, this is cross-bridge cycling, which is how the muscle will contract. So calcium will come in and bind to troponin, which will expose the binding sites on actin molecules. In turn, myosin heads will bind to the actin. The cross bridge will bend, pulling the myofilament forward. And then when the power stroke is complete, myosin will detach from actin and return to its original conformation. And the cycle just repeats as long as calcium is available. And this is an image of how this happens. And then skeletal muscle is simulated by when an action potential will travel down the motor neuron, which will release acetylcholine at the neuromuscular junction. And this acetylcholine will bind to receptors, which will trigger an action potential in the muscle fiber. And then the action potential will be propagated along the muscle fiber, and T tubules will dip into the muscle fiber, and an action potential will enter the interior. So the potential will be not just on the surface, but also interior of the muscle. And this also induces changes in adjacent sarcoplasmic reticulums. So it just spreads faster. Calcium is released by the sarcoplasmic reticulum. And then to terminate this signal, calcium will be reabsorbed back into the SR using ATPase pumps, so using active transport. And then, so here are a few examples of how. Okay, so here single twitch is a singular action potential that produces a brief contraction. So just a very simple one, a singular potential, one contraction. And then twitch summation happens when repeated action potential happens before full relaxation. So you will add the second contraction on top of the first one because the muscle didn't relax before the second contraction happened. And this is going to lead to increased force production due to additional calcium availability. And then tetanus is just sustained continuous contraction of muscle because of very high frequencies of stimulation. And then so here is 3 different types of fibers in cardiac muscle. So slow oxidative fibers are type 1, 60 to 100 milliseconds until peak tension. Lower myosin ATPase activity, so they're kind of slower, and they have very high resistance to fatigue, so they can continue to work for long periods of time. Fast oxidative fibers are type 2A, and they're 20 to 40 milliseconds until peak tension, and they have higher myosin ATPase activity, but intermediate resistance to fatigue, so they will tire out way faster. And then fast glycolytic fibers, they're type 2B, 2D, or 2X, and they're very similar to fast oxidative fibers, but instead they have very low resistance to fatigue and they work in mostly anaerobic conditions. Okay, and then muscle spindles will sense the muscle stretch, which will initiate a stress reflex. And then Golgi tendon organs, which are the tendons just attaching the muscle to the bone, they sense muscle tension and they will inhibit contraction so the muscle doesn't get damaged. And then gamma motor neurons initiate the contraction of muscular end regions in order to be able to adjust tension of the muscle spindles in turn. Okay, so then this is the last slide. Cardiac and smooth muscle, they're both controlled by the autonomic nervous system, so they're both involuntary. Their cells are connected by gap junctions, and they can both work without neural input, and they rely on calcium signaling for contraction. especially extracellular calcium. And then cardiac and skeletal muscle, they both have sarcomeres, they're both striated, and they're both going to use troponin and tropomyosin for contraction regulation. So that system I was talking about, the contractile system that keeps going in a loop. But yeah, those are the slides for chapter 8. And I know that was kind of short, but chapter 8 is kind of just going over the slides as much as you can and trying to memorize or I guess learn everything that's in there. But yeah, that's all I have. Do you guys have any questions for us?